In 1988, marine researchers documented a jellyfish that refused to follow the final step of life. Aging in complex animals moves in one direction. Cells accumulate damage, DNA shortens at its protective ends, known as telomeres, repair systems slow, and tissues gradually fail. This pattern holds across mammals, birds, reptiles, and fish. Even the longest lived whales eventually weaken. Most jellyfish are no exception. After reproducing, the adult medusa reaches the end of its cycle: birth, growth, reproduction, death. But one species did not comply. In a laboratory dish, an adult jellyfish under stress did not decay. It did not regenerate a limb. It did not produce a clone. Its body contracted inward. Tentacles were reabsorbed. Tissues reorganized. The structure of the adult form dissolved into something earlier. Within days, it returned to its polyp stage, the earliest chapter of its life cycle. Not slower aging, not extended lifespan, a reset. The species was named Turritopsis dohrnii, only a few millimeters wide, nearly transparent, found in warm coastal waters across the world. Instead of moving forward through time, it reversed direction. Scientists call the process transdifferentiation. Mature cells transform into different types, abandoning their former identity. In this jellyfish, adult cells become juvenile cells again. The medusa becomes a polyp colony, then the colony matures into a new medusa. The cycle can repeat. It can still be eaten. It can still be destroyed. Predators and disease remain, but absent external harm, biological aging does not conclude its story. Most animals travel through life along a fixed path. This one loops. But what mechanism allows an adult body to reorganize itself into youth? How do its cells rewrite their developmental fate? Why would evolution preserve a trait that appears to resist the finality shared by nearly every complex organism? Tonight, we examine how jellyfish reverses its life cycle and what that reversal reveals about the limits of aging. But before we begin, if you enjoy calm and soothing explorations like this one, filled with wonder, science, and the gentle unraveling of life's mysteries, I'd love it if you gave this video a gentle like, and maybe tap subscribe. It helps more curious minds find their way to this peaceful little corner of the internet. And if you'd be so kind, let me know in the comments, where are you winding down from tonight, and what time is it there? It's always a joy to see how far across the world this little sleepy corner of science travels. Now take a deep breath, let your shoulders soften, and together, let us move carefully into the biology of the immortal jellyfish. The word immortal often arrives wrapped in myth, as though it belongs to stone gods or distant stars. In biology, it carries a quieter meaning. It does not promise endless existence. It does not shield a body from teeth, toxins, or the slow chemistry of polluted water. Instead, it speaks only of time within the cells, of whether aging itself is allowed to complete its usual work. For most living creatures, time leaves a measurable signature. Cells divide, and with each division, small errors gather. Protective sequences at the ends of chromosomes grow shorter. Proteins misfold. Repair systems lose efficiency. Gradually, the body becomes less resilient, less capable of restoring what daily life erodes. This steady accumulation is what we recognize as aging. Eventually, it leads to decline even in the absence of predators or catastrophe. Biological immortality describes something far narrower. It refers to an organism that does not show this intrinsic, age-driven deterioration. If it avoids injury and disease, its tissues do not progress toward an internal expiration date. The clock does not strike a final hour from within. The possibility of death remains, but not because the body has simply grown old. The small transparent jellyfish introduced earlier fits within this restrained definition. It does not drift endlessly through centuries in a single delicate form. It does not remain unchanged while oceans warm and currents shift. Instead, when conditions are harsh or resources scarce, it performs a transformation that removes it from the path aging would normally follow. By returning to an earlier developmental stage, it sidesteps the terminal phase typical of adult life. The reset interrupts the usual arc from maturity to decline. This does not mean the animal lives forever automatically. A fish can swallow it in a moment. A turtle can crush it without noticing. A storm can scatter it beyond survival. Immortality in this context is conditional, dependent on circumstance. It is a capacity rather than a guarantee. Among living things, the absence of aging is not entirely unheard of. Certain bacteria divide in ways that blur the line between parent and offspring. Some single-celled protists can continue replicating without clear senescence. The freshwater hydra, a simple relative of jellyfish, maintains a population of stem cells that renew its tissues continuously, showing little measurable aging under stable laboratory conditions. These organisms share a structural simplicity. Their bodies are composed of relatively few cell types, arranged in repeating patterns that are easier to rebuild. What makes this jellyfish remarkable is that it is unmistakably an animal with differentiated tissues, nerve cells, muscle fibers, and a defined life cycle. It develops from larva to polyp to free-swimming medusa like its relatives. It possesses specialized cells that perform distinct tasks. In most animals, such specialization is a one-way commitment. A muscle cell remains muscle, a nerve cell remains nerve, and the architecture of adulthood is fixed. Here, specialization can be reversed. Mature cells relinquish their identities and participate in constructing an earlier form. Aging is not merely slowed, but rerouted. The adult body does not persist unchanged. It yields, reorganizes, and begins again. Within the language of biology, this capacity places the species in rare company. Immortal then becomes a careful word. It does not suggest indestructibility. It does not elevate a drifting invertebrate into legend. It names a specific defiance of senescence, a refusal to let internal time dictate an inevitable end. In the gentle pulse of coastal waters, that distinction is extraordinary enough. Long before a bell ever pulses through open water, the story of a jellyfish begins as a drifting union of cells, scarcely visible, suspended in currents that carry both nourishment and chance. Fertilized eggs develop into tiny larvae, known as planulae, oval and ciliated, their surfaces lined with microscopic hairs that guide them through the sea. They move not with intention, as we understand it, but with subtle coordination, propelled by rhythmic beating that steers them toward light, shadow, texture. After a brief journey, the larva settles. It attaches itself to rock, shell, or submerged surface, and its body reshapes into a polyp. This stage resembles a slender tube, anchored at one end, crowned with delicate tentacles arranged around a central mouth. Fixed in place, the polyp does not wander. It feeds on passing plankton, extending its tentacles to capture minute prey, drawing sustenance inward. In this quiet posture, it may persist for months or even years, budding clones of itself in small colonies that share the same genetic identity. When environmental cues align, temperature, food availability, seasonal rhythms, the polyp begins a transformation known as strobilation. Its body segments horizontally, forming stacked discs like a column of living saucers. Each disc gradually develops the features of a juvenile medusa called an ephyra. One by one, these young detach and rise into the surrounding water. The ephyra is a fragile geometry of lobes and canals, its symmetry already hinting at the adult form. As it grows, the lobes fill out, the bell rounds, tentacles lengthen, and internal structures refine. Muscular contractions become stronger, more coordinated. Sensory organs along the bell margin detect light and gravity, allowing orientation within the water column. The animal now occupies the Medusa stage, the form most people recognize, a drifting bell trailing threads that shimmer in filtered sunlight. In the majority of jellyfish species, this is the final act of development. The Medusa feeds, grows, and eventually reproduces, releasing eggs or sperm into the surrounding water. Fertilization occurs externally, and the cycle begins anew. After reproduction, decline follows, tissues weaken, movement slows. The body that once pulsed with measured contractions becomes less responsive. Death arrives not through dramatic rupture, but through gradual failure, the conclusion of a life that has moved steadily forward through its stages. This sequence, egg to larva, larva to polyp, polyp to medusa, defines the rhythm of most jellyfish. Each stage prepares the next. Each transformation advances in a single direction. The medusa is not a temporary costume, but the culmination of development, a final architecture shaped for dispersal and reproduction. Against this prevailing pattern, the species we follow diverges. It begins as an egg, settles as a polyp, rises as a medusa, just as its relatives do. Yet the finality that governs other jellyfish does not hold in the same way. The adult form is not an irreversible summit. Instead of standing as the endpoint of growth, it retains a latent flexibility, a capacity hidden within its tissues. Where others complete their cycle and fade, this one preserves the possibility of return, altering the quiet certainty that defines the ordinary jellyfish life. Under strain, its rhythm changes. The steady pulse that once carried it through open water softens, then falters, as though the body has begun listening to a quieter instruction written deep within its tissues. Disturbance can arrive in many forms: a sudden shift in temperature, a tear along the bell, a scarcity of drifting nourishment. Conditions that would mark the beginning of decline for other creatures become, for this small drifter, the signal for transformation. Its movement slows until it can no longer hold its place above. Gravity, subtle but constant, draws it downward. The descent is unhurried, a gradual surrender to the weight of its own substance. When it reaches the seabed, it does not struggle to rise again. Instead, it rests against the surface, translucent form, settling like a fallen petal. The change begins at the margins. Slender tentacles shorten, their length withdrawing as if reeled gently inward. Cells that once maintained their extension alter their activity, reorganizing internal scaffolds of protein. The filaments thicken, then diminish, until they are little more than faint ridges. Along the bell, muscle fibers loosen their coordinated tension. The dome that once opened and closed with measured contractions tightens slightly, its curve drawing inward. Soon, the entire body begins to contract. Not violently, not suddenly, but with deliberate patience. The umbrella shape compresses. Internal canals that carried nutrients shift their arrangement. Layers of tissue slide past one another, guided by chemical signals diffusing from cell to cell. Each microscopic instruction passes quietly, yet together they direct a remarkable restructuring. As hours pass, the animal's outline becomes simpler, less like the form it held before. The bell folds upon itself, edges meeting, surfaces touching that were once separated by open water. The organism secretes a thin coating of protective material, enclosing itself within a delicate capsule. In this state, it resembles a minute sphere, smooth and still, anchored lightly to the substrate. This phase is often described as cyst-like, though it is not a dormant seed. Within the enclosure, activity continues with intricate precision. Cells that had specialized for swimming or feeding relinquish those roles. Their internal chemistry shifts. Genes once silent become active, while others grow quiet. Structures break down and are rebuilt in new configurations. What had been muscle can be reassigned. What had been part of a sensory organ can contribute to a different tissue. The body is not repairing damage in the usual sense. It is revising its own blueprint while remaining alive. Gradually, extensions begin to emerge from the sphere's surface. These are not remnants of the former anatomy, but fresh outgrowths, slender and upright. The capsule opens, and the organism attaches itself firmly to the substrate. Its form now resembles a tiny stalk once more, crowned with delicate projections arranged around a central opening. The earlier drifting stage has given way to a rooted existence. From this renewed foundation, growth proceeds quietly. Buds appear along the sides, each capable of developing into another individual identical to the first. The colony expands by repetition, a cluster of living threads rising from a shared base. Time, which seemed poised to conclude the animal's story, has instead been redirected into a beginning that echoes an earlier chapter. Nothing in this passage suggests haste. The reversal unfolds with the same measured pace that governs tides and seasons. Where deterioration might have continued, reorganization has taken its place, and the life that once drifted freely now stands anchored, continuing in a form both older and younger at once. Identity in living tissue is usually a permanent assignment. Once a cell accepts its role, it tends to remain faithful to it for the duration of its existence. A cell that forms part of a surface layer continues as a protective tile. One that carries signals maintains its delicate branching shape. Another that contracts does so again and again, repeating its motion until it wears out. In most animals, this loyalty is enforced by chemical markings along the genome, subtle molecular tags that keep certain genes active while locking others away in silence. The effect resembles a vast library where only a few chosen volumes remain open while the rest stay sealed. Within the small creature drifting through this narrative, those volumes are not permanently closed. When its cycle turns inward, cells begin to loosen their commitments. The process is known as transdifferentiation, a word that describes a direct change from one mature form to another. Instead of returning to an unspecialized state and starting over, a cell alters course midstream, exchanging its function for a different one. The shift occurs through coordinated changes in gene expression, guided by regulatory proteins that attach to DNA and adjust which instructions are read. Entire suites of traits can be silenced while new ones emerge. Under a microscope, this transformation appears almost like a quiet rehearsal. A cell that once served as part of an outer covering gradually reorganizes its interior. Structural fibers dissolve and reassemble. The pattern of proteins along its surface changes. Electrical properties adjust. In time, that same cell may resemble one suited for transmitting signals, its chemistry aligned with a role it never previously performed. No external sculptor intervenes. The alteration arises from within, orchestrated by molecular cues exchanged among neighboring cells. Such flexibility is extraordinarily rare in animals with multiple tissue types. In mammals, birds, and reptiles, specialization is usually a one-way path. Skin remains skin, muscle remains muscle. Attempts to force a mature cell to switch identities often require artificial laboratory techniques, and even then, the process can be unstable. Complex bodies depend on predictability. Organs function because their cells hold fast to their designated tasks. Stability supports survival. Yet in this species, stability is paired with a hidden plasticity. The genome contains regulatory networks capable of reopening developmental pathways that were thought to be sealed after early growth. Genes associated with early formation, normally dormant in adulthood, become active again. Others that define the mature state recede. The transformation does not spread chaotically. It follows an orderly progression, as if an internal script guides each step with careful timing. This capacity is biologically radical because it challenges a principle long considered fundamental. Differentiation, the process that gives rise to specialized cells, has often been viewed as irreversible in complex organisms. The assumption shaped decades of biological thought. Here, that assumption softens. The boundary between one cell type and another proves more permeable than expected, at least within this lineage. Scientists studying these transformations observe that the change is not merely cosmetic. The reconfigured cells perform the duties of their new identities, contributing to tissues appropriate for an earlier life stage. Function follows form, and form follows gene activity, all unfolding within a living body that remains intact throughout the transition. What appears from afar as a simple reversal is, at the microscopic level, a symphony of molecular adjustments, each note precise, each entrance timed. In the stillness of this cellular exchange, identity becomes something fluid rather than fixed, a quality that can be rewritten without destroying the organism that contains it. The discovery did not begin with a search for immortality. It began with routine collection jars, labeled and arranged beneath fluorescent lights in a modest laboratory near the Mediterranean coast. In 1988, a young marine biologist named Christian Sommer was studying small hydrozoan jellyfish gathered from local waters. His work focused on their life cycles, the familiar progression from larval settlement to polyp and onward to the free-swimming adult stage. The animals were kept in controlled aquaria, their conditions carefully monitored, their development recorded with patient attention. At first, nothing seemed unusual. The medusae matured as expected, transparent bells drifting within glass walls instead of open sea. Then, after reproducing, several individuals failed to follow the anticipated path toward decline. Rather than disintegrating, their bodies altered in a manner that did not fit established descriptions. Sommer observed changes that suggested regression rather than decay. The forms appeared to contract and reorganize, moving not toward dissolution but toward an earlier configuration. The observation unsettled the ordinary assumptions that guided marine biology at the time. Life cycles in jellyfish were understood as directional, advancing through discrete stages. Reversal from adult to polyp was not part of accepted theory. Initial reactions leaned toward skepticism. Perhaps the specimens had been misidentified. Perhaps unnoticed larvae had settled in the tank, giving the illusion of transformation. Such possibilities required elimination before any extraordinary claim could be considered. Sommer and colleagues maintained careful records. They isolated individuals, ensuring that no hidden polyps could account for the emerging forms. They documented the sequence of structural changes under controlled conditions. Over time, repeated instances produced the same pattern. Adult medusae, subjected to stress, did not simply perish. They underwent a coordinated restructuring and reappeared as polyps attached to the tank surfaces. Scientific progress moves through replication. Other researchers began to examine the phenomenon, collecting specimens from different regions and observing them independently. Laboratories in Italy and Japan confirmed similar reversals. The species was formally described and named Turritopsis dohrnii, distinguishing it from closely related hydrozoans. As reports accumulated, the once surprising observation gained credibility through repetition. Publication followed, not in sensational headlines but within peer-reviewed journals where methodology, controls, and photographic evidence were scrutinized. Detailed descriptions outlined the stages of regression and reformation. Microscopic examinations supported the conclusion that the transformation occurred within the same individual rather than through hidden reproduction. The language of the papers remained measured, cautious, grounded in observation rather than speculation. In the years that followed, molecular techniques refined the understanding of what Summer had first witnessed. Genetic analyses confirmed species identity. Experimental manipulations demonstrated that environmental stress could reliably trigger the reversal process. What began as an unexpected anomaly in a laboratory dish evolved into a recognized biological trait supported by multiple lines of evidence. The discovery did not overturn biology overnight. Instead, it expanded the catalog of known life strategies by one quiet entry. A small hydrozoan from coastal waters had revealed a flexibility few had imagined possible in a differentiated animal. The path from observation to acceptance unfolded gradually, shaped by careful documentation and the steady patience of researchers willing to look twice at what seemed, at first glance, impossible. Hidden inside each cell rests a manuscript written in 4 chemical letters, folded so tightly that its length must coil thousands of times to fit within a space smaller than a grain of dust. For centuries, that script remained unread, its symbols visible only as faint threads under a microscope. In recent decades, new instruments have learned to separate those strands, to translate their sequences, and to arrange the letters in long digital lines. When researchers finally traced the genome of the small hydrozoan, already known for its unusual life history, they found not a single secret of immortality, but a constellation of subtle variations scattered across its genetic text. Some of these variations appear in regions associated with DNA repair. In every organism, sunlight, natural radiation, and ordinary metabolism introduce tiny injuries into genetic material. Most species possess enzymes that locate these flaws and mend them, restoring the original sequence before errors multiply. In this jellyfish, several repair-related genes show expanded activity or altered structure, suggesting that its cells maintain an especially attentive maintenance system. The proteins encoded there resemble vigilant caretakers, scanning the genome and correcting damage before it can accumulate into lasting harm. Elsewhere in the sequence lie instructions connected to telomeres, the repeating segments that cap chromosome ends. In many animals, these caps shorten with each cell division, eventually reaching a point where replication can no longer proceed safely. Certain genes in this species are linked to the preservation of those protective tips. Their patterns hint at mechanisms that stabilize telomere length, allowing cells to continue dividing without entering the usual state of exhaustion. The effect is not limitless growth, but a postponement of the internal signals that ordinarily mark cellular aging. Researchers also noted distinctive arrangements among genes that regulate stem cell behavior. Stem cells are unusual residents within a body, capable of producing many different tissue types depending on which molecular cues surround them. The genome of this jellyfish contains regulatory elements that appear especially responsive, able to activate developmental programs long after early growth has ended. These elements function like switches embedded deep within the genetic circuitry, turning on sets of instructions that in most animals remain permanently silent once maturity is reached. Comparative studies with related jellyfish species reveal how unusual this combination may be. When scientists align the sequences side by side, similarities stretch across vast portions, reflecting shared ancestry and common body plans. Yet within certain clusters of genes, differences become more pronounced. Variations in regulatory regions, small insertions or deletions, and altered expression patterns distinguish this species from its relatives that follow a more conventional lifespan. None of these differences alone explains its abilities. Together they form a network of subtle adjustments, each contributing a thread to a broader biological capacity. Genomes rarely speak in single declarations. They communicate through patterns, through the interplay of many genes acting at once. In this case, the pattern suggests a system tuned for flexibility, maintenance, and renewal. The language of nucleotides does not proclaim immortality as a headline. Instead, it offers a quiet suggestion that the processes governing repair, division, and identity remain unusually open to revision. As sequencing technologies continue to refine their resolution, additional details emerge. Small motifs, once overlooked, now visible in sharper clarity. Each newly mapped segment adds nuance to the portrait, revealing that the remarkable life cycle, observed under glass and lens, is supported by a molecular foundation, both intricate and restrained. The genome does not overturn the principles of biology. It simply arranges familiar components in an uncommon harmony, allowing a creature scarcely larger than a pinhead to carry within its cells a set of instructions unlike any yet read in the animal world. Time leaves its faint handwriting inside living cells long before it becomes visible to the eye. The marks are minute, inscribed in molecules rather than wrinkles or slowed movement. Within the nucleus of each cell, chromosomes carry strands of DNA that must be copied whenever division occurs. At the tips of these strands lie telomeres, repeating sequences that function like protective buffers. Each replication trims them slightly, not by intention but by the mechanics of copying itself. Over many cycles, the shortening becomes significant, and when the telomeres grow too brief, the cell receives a signal to stop dividing. This limit protects the organism from uncontrolled growth, yet it also establishes a quiet boundary for renewal. Alongside this gradual shortening, another influence presses steadily on cellular structures. Ordinary metabolism, the very chemistry that releases energy from nutrients, produces reactive oxygen molecules. These highly active particles drift through tissues and can collide with proteins, membranes, and DNA. The process is known as oxidative stress. Most of the time, repair systems intercept the damage. Enzymes neutralize reactive molecules, mend broken bonds, and replace injured components. Still, the work is never perfect. Small imperfections accumulate like dust in a long inhabited room, subtle at first, then increasingly difficult to clear away. DNA itself bears the record of these encounters. Radiation from natural sources, stray chemical reactions, and simple copying errors introduce tiny alterations into the genetic code. Specialized repair pathways patrol the genome, scanning for mismatches or breaks. When they find a flaw, they cut, replace, and reseal the strand. Yet no surveillance system operates without limits. Some injuries escape correction, and each uncorrected change becomes part of the cell's permanent script. Over years, these alterations can disrupt normal function, weakening tissues, or altering the signals that guide growth. In most animals, these processes unfold together, telomeres shortening, oxidative effects accumulating, DNA acquiring scattered changes. The body continues to function because countless cells divide, replace, and compensate for one another. Eventually, though, the balance shifts. Renewal cannot keep pace with loss. Organs become less efficient, responses slower, structures more fragile. Aging emerges not from a single cause, but from this layered convergence of molecular events. The remarkable species at the center of this story moves through the same biochemical world as any other creature. Its molecules obey the same physical laws, its cells face the same threats. What distinguishes it is not an absence of damage, but an unusual relationship with it. When its life cycle turns upon itself, cellular states that would normally be fixed become flexible. Genetic programs associated with early development can be reactivated, allowing tissues to reorganize rather than simply deteriorate. The molecular wear that accumulates does not vanish, yet it can be diluted or redistributed as new structures form from existing material. For larger and more complex animals, such reversal is constrained by architecture. Bodies composed of many specialized organs depend on stable organization. A heart must remain a heart, a lung, each part synchronized with the others. To dismantle and rebuild such systems would require coordination across billions of cells, each maintaining precise timing. Even slight errors could disrupt essential functions. Evolution has favored reliability over reversibility in these lineages, preserving forms that persist steadily rather than those that risk transformation. Thus, aging proceeds along its familiar course in most creatures, guided by the chemistry of replication and repair. The slow shortening of telomeres, the steady influence of oxidative reactions, and the accumulation of genetic alterations together shape the arc of life. Within that broad pattern, rare exceptions appear, not by escaping biology's rules, but by arranging them differently, allowing time's handwriting to be revised before it fully settles into permanence. In the human body, complexity is layered like an intricate city built over centuries, each district assigned a purpose and bound to the others through countless lines of communication. From early development onward, cells follow narrowing paths. A cluster destined to become heart tissue begins expressing one set of genes, while neighboring cells commit to forming bone, blood, or brain. Chemical signals reinforce these decisions, adding molecular markers to DNA that silence alternative possibilities. With time, the options fade. Identity becomes deeply inscribed. This specialization grants extraordinary capability. A neuron can transmit electrical impulses across vast cellular distances. A cardiac muscle cell can contract in steady rhythm for decades. Liver cells process toxins and synthesize vital proteins with remarkable efficiency. Yet this precision depends on stability. Each cell type maintains its function by keeping large portions of its genetic library locked away. Reopening those volumes risks disturbing the delicate balance required for organ performance. In rare laboratory settings, scientists have learned to coax adult human cells back toward a more flexible state. By introducing specific transcription factors, they can create induced pluripotent stem cells, which regain the capacity to differentiate into many tissue types. Even under controlled conditions, this reprogramming carries hazards. Cells that divide rapidly and shed their specialized restraints can begin to grow in uncontrolled ways. Tumors arise when regulatory systems fail to contain proliferation. The same plasticity that enables regeneration can also seed malignancy. Cancer reveals how thin the line can be between renewal and disorder. Mutations that activate growth, promoting genes, or disable growth, suppressing genes, allow cells to multiply without regard for surrounding tissues. In a complex organism composed of trillions of cells, such rogue behavior threatens the integrity of the whole. Evolution has therefore favored mechanisms that limit excessive flexibility. Checkpoints monitor DNA integrity. Telomere shortening restricts indefinite division. Programmed cell death removes damaged cells before they expand uncontrollably. These safeguards reduce the risk of catastrophic growth, though they also constrain the potential for radical transformation. The small hydrozoan described earlier operates under a different scale of organization. Its body plan lacks the layered organ systems that define mammals. It possesses no centralized brain, no intricately partitioned heart, no lungs exchanging gases within branching airways. Its tissues are simpler, arranged in fewer types with more direct interactions. When its cells alter identity, the coordination required is substantial, but not comparable to dismantling and rebuilding a vertebrate body with dozens of interdependent organs. Evolution shapes traits not according to possibility alone, but according to advantage within a given environment. For large, long-lived animals, maintaining structural continuity may confer greater survival than the ability to revert to an earlier developmental state. A stable body capable of sustained movement, complex behavior, and parental care offers benefits that outweigh the rare chance of complete cellular reorganization. The cost of such stability is aging, an accumulation of molecular changes that cannot be entirely undone without risking systemic collapse. Trade-offs permeate biology. Energy invested in rapid growth may reduce investment in repair. Robust immune defenses may increase inflammation over time. In humans, extended lifespan already exceeds that of most mammals of similar size, supported by intricate regulatory networks that balance regeneration and restraint. To introduce wholesale reversal of differentiation into such a system would require re-engineering these networks in ways that might destabilize the protections currently in place. Thus, the inability to reset, as that hydrozoan does, is not merely a missing switch waiting to be flipped. It reflects a deeply integrated architecture shaped by millions of years of selection for coherence, coordination, and controlled growth. Our cells hold vast potential within their genomes, yet that potential is governed by boundaries that preserve the whole organism. Within those boundaries, life unfolds forward, sustained by renewal but anchored by form. Along shifting coastlines where currents braid together and tides redraw the margins each day, survival favors those who can adapt quickly or endure loss without final consequence. The small hydrozoan at the center of this story occupies such restless waters. It is minute, nearly weightless, easily carried beyond the place where it first formed. Its size allows it to slip through narrow gradients of temperature and salinity, yet that same smallness renders it vulnerable to almost everything larger than itself. In these coastal zones, predation is constant. Larval fish feed on drifting plankton. Crustaceans sweep the water with feathery appendages. Other gelatinous animals pulse nearby, consuming whatever fits within their reach. For a creature measured in millimeters, concealment is limited and defense minimal. Many individuals never complete even a single reproductive cycle. Under such pressure, longevity through sheer durability would be unlikely to evolve. Instead, resilience may emerge through repetition. The ability to reverse development offers a subtle advantage in this context. Injury that would normally end a life can become a signal to reorganize. If a medusa suffers partial damage yet retains sufficient cellular integrity, reverting to a more resilient form anchored to a substrate may provide a second opportunity. The polyp stage, with its simpler architecture, and capacity for clonal budding can generate multiple new medusae over time. From one individual under stress, a small colony may arise, increasing the odds that at least some descendants will drift into favorable conditions. Coastal environments are not only biologically crowded, but physically unstable. Storms churn sediments and alter light penetration. Freshwater runoff from rivers changes salinity within hours. Seasonal shifts modify plankton abundance, reshaping the entire food web. A life cycle that includes both a mobile phase and a sessile phase allows exploitation of different niches. When the water column proves inhospitable, attachment to a solid surface may provide refuge. When currents become rich with prey, the medusa form can disperse and feed. Restarting development also spreads risk across time. Rather than investing entirely in a single trajectory toward reproduction, followed by decline, this species can intersperse phases of growth and renewal. Genetic material persists not only through offspring, but through repeated cycles within the same lineage. In evolutionary terms, what matters is the transmission of genes. If reversal increases the number of reproductive events an individual lineage can achieve, natural selection may favor the underlying mechanisms, even if many individuals still perish from predation. Small body size further amplifies this strategy. The metabolic demands of a tiny organism are relatively modest. Reorganizing tissues requires energy, yet the scale is manageable. In larger animals, the energetic cost of dismantling and reconstructing complex organs would be immense and potentially fatal. Here, the simplicity of structure reduces that burden. The transition from drifting medusa to rooted polyp involves fewer cell types and less intricate coordination than a comparable shift would in a vertebrate. Over generations, such flexibility may stabilize population numbers in environments where mortality is unpredictable. Even if waves of predation remove many medusae, surviving polyps attached to submerged surfaces can quietly regenerate the free swimming stage when conditions improve. This alternating pattern creates a buffer against sudden environmental swings. Evolution does not design with foresight. It preserves traits that confer relative advantage within specific contexts. In the variable margins of warm seas, a life capable of folding back upon itself may outlast one bound to a single forward path. The capacity to begin again, not as an abstract ideal but as a biological function, fits the rhythms of places where change is the only constant. Across belts of warm ocean that circle the planet like soft ribbons, this small hydrozoan lives where temperature remains gentle and seasonal extremes rarely intrude. Such waters lie near tropical and subtropical coasts, where sunlight filters deeply and microscopic life flourishes in quiet abundance. Within these regions, the animal drifts among particles invisible to the casual eye, occupying a realm measured not in miles but in centimeters, where the presence of nourishment depends on subtle shifts in current and light. Its distribution is not confined to a single shoreline or basin. Records have traced it through the Mediterranean, the Caribbean, the Western Pacific, and other temperate to warm seas. The pattern suggests a traveler that has crossed oceans without intention. Part of this wide range is owed to the natural movement of water itself. Ocean currents form vast circulatory pathways, transporting plankton, larvae, and drifting organisms across great distances. Carried within these slow rivers, the species can appear far from its origin, arriving as quietly as a grain of sand settling after a long journey. Another pathway has aided its spread, one shaped by human industry rather than tides. Large ships take on ballast water to maintain stability during voyages, drawing seawater into internal tanks before crossing open oceans. When that water is released at distant ports, it carries with it minute inhabitants from the place of departure. Among these travelers may be small jellyfish, or their earlier stages, unseen passengers delivered into foreign harbors. Over decades, such transfers have introduced the species to regions it might never have reached through currents alone, allowing it to establish new populations wherever conditions resemble those of its native range. Wherever it settles, food must be present, and its sustenance consists of organisms so small they drift as part of the planktonic community. Tiny crustaceans, protozoans, and larval forms of other marine species become its nourishment. These prey items are abundant in nutrient-rich coastal zones, especially where sunlight supports blooms of phytoplankton that form the base of the food web. The hydrozoan feeds by capturing these microscopic bodies and directing them inward, converting their substance into energy for growth and maintenance. The abundance of plankton is rarely constant. Seasonal changes, shifting winds, and upwelling currents can increase or diminish concentrations within days. In some periods, the water teems with life, each drop containing a constellation of edible particles. At other times, the same water may appear clear and sparse, its resources temporarily reduced. The species persists by existing within this variability, its distribution waxing and waning with the availability of food and the movement of currents. Because it is so small, its relationship with the ocean is intimate. Large animals experience the sea as an environment through which they move. This organism experiences it as a surrounding medium that shapes every aspect of existence. Temperature changes of a few degrees, salinity shifts from rainfall or evaporation, and subtle chemical differences all influence its survival. Its presence in any given location reflects a delicate balance between these factors, a balance maintained only while conditions remain within tolerable limits. Over time, mapping studies have revealed a patchwork pattern rather than a continuous spread. Populations appear in scattered clusters, often near coasts where shipping activity is frequent and plankton productivity is high. Between these clusters lie stretches of ocean where the species is seldom recorded, perhaps absent, perhaps simply unnoticed. Its global presence is therefore both wide and fragmented, like a constellation whose points shine softly across distant waters, connected not by land but by the slow and patient motion of the sea. In the quiet arithmetic of the sea, survival is never granted by a single remarkable trait. Every organism exists within a network of appetites, movements, and encounters that shape its chances from moment to moment. The small hydrozoan that can renew its life cycle is no exception. Its unusual biology alters its relationship with time, yet it remains part of a living community where many mouths open and close in search of nourishment. Fish are among its most frequent consumers. Juvenile reef species, drifting larvae, and small pelagic hunters feed opportunistically on gelatinous prey. To them, the tiny medusa is not a marvel, but a morsel, soft-bodied and easily swallowed. Vision in many fish is tuned to detect faint contrasts and motion, allowing them to notice delicate forms suspended nearby. A quick intake of water, a brief expansion of the jaws, and the organism vanishes into a digestive tract where enzymes dismantle its tissues into usable molecules. Sea turtles also participate in this exchange. Some species specialize in gelatinous diets, their throats lined with backward-pointing papillae that guide slippery prey toward the stomach. Though these reptiles often target larger jellyfish, they do not ignore smaller drifting forms encountered along their paths. Each feeding pass removes individuals that might otherwise have continued their cycles. The turtles do not distinguish between ordinary jellyfish and those capable of unusual reversals. To a predator, nutritional value matters more than biological novelty. Even among gelatinous animals themselves, predation occurs. Larger jellyfish capture smaller ones with trailing tentacles armed with stinging cells. Contact triggers microscopic harpoons that inject toxins, immobilizing the prey before it is drawn inward. In these moments, the boundary between species dissolves into a simple transfer of energy, one body sustaining another. The ocean hosts countless such exchanges, each a small transaction in the larger economy of life. These interactions reveal an important truth about biological immortality. The term describes resistance to aging, not freedom from hazard. An organism may possess the capacity to reset its developmental state, yet still be consumed long before that capacity is used. Predation acts swiftly, often without warning. Teeth, venom, and digestive enzymes operate on timescales far shorter than cellular reorganization. A remarkable life history cannot prevent an encounter that ends in an instant. Because of this, the species does not dominate its environment despite its unusual abilities. Its populations remain modest, balanced by the many forces that remove individuals as quickly as they arise. Ecological success depends on more than longevity. It depends on growth rate, reproductive output, mobility, and resistance to predators. In some of these measures, the small hydrozoan is unremarkable. It drifts rather than swims with strength, feeds on tiny prey, and offers little physical defense. The ocean favors equilibrium. Predators limit the abundance of prey, while prey availability shapes predator numbers. Within this balance, no single adaptation guarantees supremacy. The capacity to renew life stages may help a lineage persist through adversity, yet it does not exempt it from the shared rules that govern all marine creatures. Each individual remains subject to chance encounters, shifting conditions, and the ever-present search for food carried out by others. So the animal's unusual relationship with time exists alongside very ordinary vulnerabilities. It can revise its internal clock, yet it cannot step outside the wider web in which it lives. Around it, fins slice through water, shells glide silently, and translucent hunters trail their threads. The currents that carry nourishment also carry danger, and within those currents, the small voyager continues its brief passages, sustained for as long as circumstance allows. Among the many forms life has taken on this planet, a few possess an unusual relationship with injury and time, as though their bodies remember possibilities long set aside in others. The small hydrozoan described earlier belongs to this rare company, yet it is not alone in displaying biological flexibility. Other simple organisms also carry traces of regenerative talent, each expressing it in ways shaped by their own structure and history. Consider the freshwater hydra, a tiny relative within the same broad phylum. Its tubular body is composed of only a few tissue layers, constantly renewed by populations of stem cells that divide throughout its life. In stable conditions, hydra show little sign of senescence. Cells are replaced before they deteriorate, and tissues maintain their function without the gradual decline typical of many animals. If cut into pieces, fragments can reorganize and grow into complete individuals, each part restoring the symmetry of the whole. This capacity arises from the persistence of pluripotent cells distributed throughout its body, ready to differentiate into whatever type is required. Flatworms offer another example. Certain species, especially planarians, can regenerate entire bodies from small sections. When divided, Each fragment rebuilds missing structures through coordinated cell division and pattern formation. Within their tissues reside neoblasts, versatile cells capable of producing every other cell type. These cells respond to injury by proliferating and migrating to damaged regions, reconstructing organs with remarkable accuracy. Such regeneration has fascinated biologists for centuries, revealing how developmental instructions remain accessible even in adulthood. Yet, neither Hydra nor flatworms perform the specific transformation seen in the Hydrozoan we have been following. Hydra maintain a steady state of renewal, replacing parts without reversing their overall developmental stage. Flatworms rebuild lost structures, but they do not routinely transform from an adult body back into a juvenile form as part of their normal biology. Their abilities restore what has been removed, rather than redirect the entire life cycle. The distinction lies in direction. Most regenerative organisms repair or replace while continuing forward through time. Their development remains linear, even when interrupted by injury. The Hydrozoan's ability is different in kind. It can shift from a mature state to an earlier configuration, reorganizing its tissues so thoroughly that the sequence of its life can begin again. This is not simply regrowth but reversion, a coordinated return that alters the temporal order of its own existence. Part of what allows this lies in its structural simplicity. Like Hydra, it possesses relatively few cell types compared with complex animals. This simplicity reduces the number of signals that must be synchronized during transformation. At the same time, its life cycle includes distinct stages adapted to different ecological roles, which means its genome already contains instructions for multiple body plans. The remarkable feature is that these instructions remain accessible even after one stage has been completed. Comparative genetic studies suggest that while many organisms retain dormant developmental pathways, most lack the regulatory flexibility to reactivate them once maturity is reached. In the Hydrozoan, certain molecular switches appear unusually responsive, permitting the reassembly of earlier forms when conditions demand it. The presence of such switches does not make it the only regenerative creature, but it does place it at an unusual intersection between regeneration and life cycle reversal. Thus, within the quiet catalog of living strategies, it stands neither entirely alone nor easily matched. Others renew tissues, others rebuild lost parts, yet this species combines renewal with a return to youth, altering not just structure but sequence. In that subtle difference rests the reason it is often singled out, a small organism distinguished not by size or strength, but by the rare direction in which its life is able to flow. Every transformation in biology carries a cost measured not in currency, but in energy, the quiet fuel that powers each chemical reaction within living tissue. Even the smallest alteration in form requires molecules to be assembled, bonds to be broken, and signals to be transmitted from one cell to another. For the small Hydrozoan whose life can fold back upon itself, the return to an earlier stage is not a simple retreat, but a process that demands careful expenditure from its internal reserves. Metabolism supplies this expenditure. Nutrients gathered from prey are converted into adenosine triphosphate, the molecular unit that stores and transfers energy for cellular work. When tissues reorganize, this stored energy is consumed rapidly. Proteins must be dismantled and rebuilt. Membranes must be reshaped. Genetic instructions must be copied and expressed in new patterns. Each of these steps draws upon chemical stores accumulated during periods of feeding. Without sufficient reserves, such extensive restructuring would stall before completion. Because of this requirement, reversal does not occur constantly. The organism does not cycle endlessly between forms in ordinary conditions. Instead, it continues along its typical developmental path until circumstances shift. Environmental stress, physical damage, or scarcity may prompt the transition, yet even then the process unfolds only if enough energy remains to support it. A severely weakened individual lacking resources may not complete the transformation. The ability exists as a potential, not an automatic reflex. Observations in controlled settings suggest that individuals often resume growth in their current stage when conditions remain stable. The mature form can persist, feeding and reproducing without reverting. Only when external pressures threaten survival does the internal machinery redirect its efforts. In this sense, the transformation resembles a contingency plan embedded within the organism's biology, activated when continuation in the present state becomes less favorable than beginning anew. Such strategic use reflects a balance shaped by natural selection. Energy invested in rebuilding cannot simultaneously support reproduction or movement. If reversal were triggered too easily, individuals might abandon productive stages prematurely, reducing their chances of passing on genetic material. If triggered too rarely, they might perish before employing the mechanism. Over generations, selection would favor regulatory systems that initiate the process under conditions where the benefits outweigh the metabolic expense. The chemistry of this decision unfolds through signaling molecules that circulate among cells. These compounds respond to changes in temperature, nutrient availability, and physical integrity, altering gene activity in ways that guide the organism toward either persistence or reorganization. The signals do not possess intention, yet their interactions create outcomes that appear purposeful, as though the body were evaluating its circumstances and choosing a path. In truth, it is the cumulative effect of molecular thresholds reached and crossed. Energy itself is neither abundant nor scarce by nature. It exists in gradients, moving through ecosystems as organisms consume and are consumed. For a creature so small, the margin between surplus and deficit can be narrow. A period of plentiful feeding may fill its tissues with stored compounds, enabling the possibility of transformation if needed. A stretch of scarcity may leave it without the reserves required for such an undertaking. Thus, the remarkable ability to reset does not free the organism from ordinary constraints. It remains governed by the same energetic principles that shape all life. Reversal is possible, yet it is costly. Renewal can occur, yet it must be financed by prior nourishment. Within this balance lies the quiet discipline of its biology, a system that allows renewal not as a constant state, but as a carefully timed response, sustained only when sufficient energy has been gathered to carry it through. Numbers in nature rarely expand without restraint. Even organisms capable of unusual renewal exist within boundaries shaped by forces that operate quietly yet persistently. The small hydrozoan whose life can begin again might seem, in theory, capable of multiplying until its kind filled every suitable stretch of sea. Yet the waters remain far from crowded with its presence, and its populations appear only in scattered pockets rather than in vast, uninterrupted swarms. Part of this restraint arises from the many mouths that feed within marine ecosystems. Predators remove individuals at every stage of existence. Minute drifting forms are consumed by plankton feeders, while larger ones fall to animals adapted to gelatinous prey. Each act of feeding subtracts from the total, preventing unchecked accumulation. In ecological systems, consumption and reproduction often move in tandem, one increasing numbers, the other reducing them, creating a shifting equilibrium rather than a steady rise. Environmental pressures add another layer of limitation. Temperature fluctuations, variations in salinity, and changes in nutrient availability can alter survival from one week to the next. A region that supports growth during one season may become inhospitable during another. Storm-driven mixing can disperse organisms into unsuitable waters. Periods of low plankton abundance can leave individuals without enough nourishment to sustain themselves. These conditions do not need to eliminate a population entirely to shape its size. Even small reductions repeated over time can keep numbers modest. Reproductive biology also plays a role. Although the species can repeat stages of its life, successful continuation still depends on opportunities to produce offspring and on those offspring surviving long enough to develop. Many eggs and larvae never reach maturity, carried into unfavorable conditions, or consumed before they can settle. The potential for renewal within an individual does not guarantee success for its descendants. Each generation begins again within the same uncertain surroundings. Space itself can impose limits. Suitable surfaces for attachment are not infinite. Competition for these sites occurs among many organisms seeking footholds, including algae, barnacles, and other sessile invertebrates. When available substrate is scarce, fewer individuals can establish the anchored phase of their existence. Without that stage, certain forms of reproduction cannot proceed. In this way, the physical structure of the habitat quietly regulates abundance. Chemical factors may influence distribution as well. Coastal waters can carry pollutants, shifts in acidity, or altered oxygen levels resulting from natural cycles or human activity. Such changes affect delicate organisms quickly, sometimes preventing growth or survival even when food is present. The tolerance range for these conditions is not limitless, and populations may diminish when parameters drift beyond it. All these influences interact rather than act alone. Predation may be intense in one location but mild in another. Nutrients may be plentiful where predators are numerous, or scarce where predators are few. The resulting pattern resembles a mosaic, with clusters of individuals appearing where circumstances briefly align. Over time, currents redistribute them, and the arrangement changes again. The species' unusual capacity to renew itself does not override these broader ecological dynamics. It functions within them, offering resilience but not dominance. Survival remains contingent on the same interplay of consumption, competition, and environment that shapes every marine population. The sea is vast, yet its living communities are governed by balances that keep even remarkable organisms within modest bounds, their numbers rising and falling in quiet accordance with the conditions that surround them. Scale changes everything in biology. A transformation that unfolds smoothly within a tiny body can become almost unimaginable when multiplied across billions or trillions of cells arranged into intricate systems. The hydrozoan, whose life canny expanded into cycles of renewal, exists at a level of organization where tissues are relatively simple and communication distances are short. In such a body, signals travel quickly, instructions spread with little delay, and structural rearrangements can proceed without disrupting vast networks of interdependent parts. Larger animals inhabit a different architectural reality. Their organs are specialized districts linked by blood vessels, nerves, and chemical messengers that must remain precisely synchronized. The heart depends on rhythmic signals from pacemaker cells. The lungs rely on coordinated muscle movements and delicate membranes for gas exchange. The liver processes nutrients while filtering toxins, its functions intertwined with those of the digestive tract and circulatory system. Each organ performs tasks that support all others. Altering one structure requires adjustments throughout the entire organism, or stability falters. For a creature of such complexity to revert to an earlier developmental state, every organ would need to reorganize simultaneously while continuing to sustain life. Blood would still need to circulate, oxygen would still need to reach tissues, and neural signals would still need to coordinate movement and sensation. Even a brief interruption in these processes could prove fatal. In small and structurally simple organisms, temporary suspension of certain functions may be survivable. In large animals, continuity is essential. The nervous system presents an especially formidable barrier. In vertebrates, billions of neurons form intricate circuits that encode memory, perception, and learned behavior. These cells establish precise connections during development, guided by molecular cues that ensure each axon reaches its correct destination. Once established, these networks remain largely fixed. Reversing development would require dismantling and reconstructing these connections without losing essential function. The sheer number of synapses and their delicate specificity makes such reorganization extraordinarily difficult to achieve without catastrophic disruption. Developmental timing also complicates the picture. In embryos, tissues form in carefully orchestrated sequences, with each stage preparing conditions for the next. Hormones, growth factors, and gene regulatory networks interact in elaborate cascades. To retrace this sequence in reverse within a mature body would demand that every step be precisely coordinated in opposite order, all while maintaining the organism's viability. The probability of errors would increase with each additional layer of complexity. Evolution tends to favor solutions that enhance survival within existing constraints rather than those that require wholesale redesign. In large animals, longevity can be improved through gradual refinements such as enhanced DNA repair, more efficient immune responses, or slower metabolic rates. These strategies extend lifespan without necessitating radical restructuring of the body plan. A complete return to an earlier developmental stage might offer theoretical benefits, yet the structural and physiological challenges would be immense. The hydrozoan's capacity for renewal arises from a body built with flexibility as a foundational trait. Its tissues are arranged in ways that permit reorganization without dismantling essential systems, and its functions do not depend on the continuous operation of complex organs. This simplicity allows biological pathways that would be destabilizing in larger organisms to operate safely within its scale. Thus, the prospect of such a trait evolving in large animals is constrained not by a single missing gene, but by the architecture of complexity itself. The more elaborate the body, the more tightly its parts must cooperate, and the less room there is for sweeping transformations. Within the vast diversity of life, each form carries its own possibilities and limits, shaped by the balance between structure and change that evolution has refined over countless generations. Within every living body lies a delicate balance between growth and restraint. Cells must divide often enough to repair tissues and maintain function, yet not so often that order dissolves into excess. This equilibrium is guarded by networks of genes that encourage division when needed and halt it when limits are reached. When those controls falter in most animals, the result can be cancer, a condition in which cells multiply without coordination, ignoring signals that normally keep them aligned with the needs of the organism. The process of cellular reprogramming, in which a mature cell changes identity, shares certain molecular features with this uncontrolled growth. Both involve shifts in gene activity, reopening regions of DNA that are usually kept silent, Both require alterations in the proteins that regulate division and specialization. In human medicine, scientists who attempt to reprogram adult cells in the laboratory often observe that the same pathways associated with tumor formation become active during the transition. It is as though the cell must briefly loosen the safeguards that maintain its identity in order to assume a new one. This resemblance has long suggested that large animals rarely evolve natural mechanisms for extensive reprogramming. The danger is not simply that cells might change roles, but that they might continue dividing beyond what the body can support. A tissue composed of rapidly multiplying cells can crowd out neighboring structures, disrupt organ function, and drain resources. For complex organisms, strict regulation is essential. The genetic systems that prevent cancer also limit the degree to which cells can revert to earlier developmental states. The small hydrozoan described earlier appears to navigate this boundary with unusual precision. During its transformation, cells relinquish their specialized roles and adopt new ones, yet they do so in a coordinated pattern rather than an unchecked expansion. Studies suggest that its genome includes regulatory sequences capable of activating developmental genes while still maintaining control over division rates. Instead of unleashing growth indiscriminately, the process unfolds as a guided reorganization, with signals passing among cells to ensure that each step proceeds in harmony with the rest. Part of this control may arise from the relative simplicity of its body plan. With fewer cell types and less structural layering, there are fewer opportunities for conflicting signals to arise. Communication between cells can spread rapidly across its tissues, aligning their behavior before instability develops. Molecular pathways that in other animals might trigger tumor formation seem, in this species, to be integrated into a broader regulatory framework that channels their activity toward constructive ends. Researchers studying these mechanisms hope to understand how such regulation is achieved. If scientists can identify the genetic switches that allow cells to change identity without losing control of division, they may gain insight into how to encourage safe regeneration in human tissues. The goal is not to replicate the entire life cycle of this organism in larger bodies, but to learn from the principles that govern its transformations. By examining how its cells coordinate reprogramming while avoiding disorder, investigators seek clues that might one day inform therapies for injuries or degenerative diseases. The connection between reprogramming and cancer thus forms a quiet bridge between marine biology and medicine. On one side lies a small creature whose cells rearrange themselves as part of its natural history. On the other side stand laboratories where researchers attempt to guide similar processes under controlled conditions. Between them flows a shared question of regulation, of how growth can be permitted without becoming destructive. In the patient unfolding of this hydrozoan's transformations, science glimpses a living example of that balance, a system in which change proceeds without chaos and renewal unfolds within the bounds of order. In research centers far from any shoreline, beneath steady lights and the quiet hum of incubators, scientists examine tissues that do not belong to the sea yet are linked to it by curiosity. The small hydrozoan, whose life can turn inward, has become an unexpected reference point in conversations about healing and renewal. Its transformations are not studied as curiosities alone, but as living examples of biological processes that medicine hopes to understand more deeply. Regenerative medicine seeks ways to repair damaged tissues in the human body, to restore function after injury or disease. In certain organs, such as the skin and liver, regeneration already occurs to a limited extent. In others, including the heart and nervous system, the capacity is far more restricted. Researchers aim to uncover methods by which mature cells might be encouraged to divide safely or adopt new roles without destabilizing the surrounding tissue. Observing an organism that naturally reassigns cellular identity provides a model of how such changes can be coordinated without descending into disorder. Stem cell research lies at the heart of this effort. Stem cells possess the ability to differentiate into multiple cell types, serving as reservoirs for repair. In humans, embryonic stem cells and induced pluripotent stem cells demonstrate remarkable flexibility in laboratory settings. Yet guiding these cells toward specific fates within a living body remains complex. Signals must be precisely timed, and proliferation must be carefully regulated. The hydrozoan's life cycle suggests that regulatory networks can exist which reopen developmental programs long after maturity, then close them again once reorganization is complete. Understanding how these networks are structured may illuminate new strategies for directing stem cell behavior in therapeutic contexts. Another area of interest involves DNA repair mechanisms. Aging and many diseases are associated with accumulated genetic damage. Cells that maintain robust repair systems tend to function more reliably over time. Studies of the hydrozoan's genome have highlighted genes associated with efficient correction of DNA errors and stabilization of chromosome ends. By comparing these genes with their counterparts in humans, researchers hope to identify pathways that could be enhanced or mimicked to improve cellular resilience. Even modest improvements in repair efficiency might reduce the progression of age-related disorders. The goal is not to replicate the organism's entire life strategy within human tissues. The structural and ethical challenges of such an endeavor would be immense. Instead, scientists focus on extracting principles. How are growth signals modulated so that cells change identity without proliferating uncontrollably? How are developmental genes reactivated in a coordinated sequence? How are damaged components replaced while preserving overall integrity? Each of these questions guides experiments that extend from marine biology into clinical research. Interdisciplinary collaboration has grown around these themes. Molecular biologists sequence genes and analyze regulatory regions. Developmental biologists map signaling pathways that control differentiation. Clinicians consider how insights might translate into treatments for degenerative diseases, spinal cord injuries, or organ failure. The hydrozoan itself remains small and distant from hospital wards, yet its biology contributes to a broader understanding of how cells might be persuaded to renew themselves. In this way, the study of a drifting marine invertebrate becomes part of a larger narrative about healing. The quiet reversal of its life stages offers more than a scientific curiosity. It provides a window into the flexibility embedded within living systems, a reminder that even well-established cellular identities can, under certain conditions, be revised. Within laboratories, that reminder guides careful experimentation, grounded not in fantasy, but in the patient unraveling of mechanisms that already exist in nature. Aging can be understood as a gradual drift toward disorder, a quiet unfolding of entropy within living systems. In physics, entropy describes the tendency for organized structures to move toward less ordered states over time. In biology, this tendency appears in the slow accumulation of molecular irregularities, the subtle loss of precision in processes that once operated with effortless coordination. Cells replicate with slightly reduced fidelity, proteins fold with marginally less accuracy, and repair systems correct damage with diminishing efficiency. None of these changes is abrupt, yet together they trace a gentle slope along which living bodies travel. From an evolutionary perspective, Longevity is not always the primary objective. Natural selection favors traits that increase the likelihood of genes being passed to the next generation. If reproduction occurs successfully, the continuation of genetic material is secured even if the organism itself does not persist much longer. Many species therefore invest heavily in early growth and reproductive capacity, channeling energy into producing offspring rather than maintaining tissues indefinitely. This allocation reflects a balance shaped by environmental pressures, predation, and resource availability. A body that lasts long enough to reproduce may fulfill its evolutionary role without needing to endure far beyond that point. The biological mechanisms underlying aging reflect this emphasis. Maintenance systems exist in all organisms, yet they are calibrated to operate within limits. Perfect repair would require vast energy, and energy must also support movement, feeding, and reproduction. Evolution tends to favor sufficiency rather than perfection. As long as an organism survives long enough to reproduce, additional longevity offers diminishing returns in terms of genetic success. Over generations, this principle shapes life histories across the living world, producing patterns in which growth, reproduction, and decline follow one another in familiar sequence. Within this framework, the small hydrozoan described earlier presents an intriguing variation. Its life cycle includes the possibility of returning to a previous stage, effectively interrupting the usual progression from maturity to deterioration. This capacity does not eliminate entropy, for its molecules remain subject to physical laws, yet it alters how entropy manifests. Instead of allowing accumulated irregularities to continue building within a single form, the organism reorganizes itself, redistributing components and renewing structures. The result is not a timeless body, but a recurring opportunity for restoration. Such a strategy suggests that aging need not be expressed in only one way. In most animals, the path is linear, with change moving steadily forward. In this species, the path can curve. The existence of that curve does not overturn evolutionary principles, yet it expands the range of possibilities they permit. Natural selection has shaped a lineage in which renewal can coexist with reproduction, each supporting the other under certain conditions. The trait persists because it contributes, in its own ecological context, to the continuation of genetic material. Philosophically, this invites reflection on how life negotiates the passage of time. Aging is often viewed as an inevitable decline, a gradual surrender to disorder. Yet in nature, different organisms engage with entropy in distinct ways. Some resist it through constant cellular replacement. Others endure it until reproduction is complete. This hydrozoan redirects it, transforming structural change into an opportunity for beginning again. Its existence does not promise escape from the universal tendencies of matter, but it illustrates that biological systems can shape those tendencies into varied patterns. In the quiet rhythms of its transformations, the concept of aging appears less like a fixed destiny and more like a spectrum of possibilities, each shaped by the interplay between physical law and evolutionary design. Memory, in most animals, is not a single thing but a pattern etched into networks of cells. It resides in the strengthening of connections between neurons, in the chemical adjustments that alter how signals travel across synapses. Experiences leave traces through subtle shifts in electrical responsiveness and molecular composition. Over time, these traces accumulate into what we recognize as learned behavior, familiarity, and habit. The small hydrozoan, whose life can reverse, possesses a nervous system, though it is far simpler than that of vertebrates. In its free swimming stage, nerve cells form a loose net distributed through the body, coordinating contractions and responding to light or touch. There is no centralized brain, no layered cortex storing detailed representations of the world. Yet even a simple nerve net can encode basic forms of responsiveness. Repeated stimulation may alter how strongly the organism reacts, and coordinated patterns of movement suggest a rudimentary integration of sensory input. When reversal begins, this network does not remain intact. As tissues reorganize, nerve cells lose their established arrangements. The structures that once transmitted impulses across the body are dismantled along with other specialized tissues. Proteins that maintain synaptic connections are broken down. Cellular extensions retract. The coordinated electrical patterns that governed swimming dissolve as the organism transitions toward its earlier form. By the time the anchored stage is reestablished, the nervous system has been simplified dramatically. Polyps possess only a minimal nerve net suited to their stationary existence. They respond to contact and chemical cues, extending tentacles when prey approaches, retracting when disturbed. Their neural architecture supports these basic behaviors, but does not preserve the configuration that existed during the mobile stage. The reassembled tissues reflect the needs of the present form rather than the history of the previous one. Because memory depends on stable patterns of neural connectivity, the breakdown of that network implies the loss of any stored information. Without preserved synapses or enduring electrical circuits, there is no substrate on which past experiences could remain encoded. Each time the life cycle returns to its earlier stage, the nervous system is built anew according to developmental instructions, not reconstructed from a blueprint of prior connections. Even when the medusa stage arises again from the polyp colony, its nerve net develops as part of normal growth. The emerging structure resembles that of its predecessors, but is not a continuation of a specific individual's neural history. It is a fresh arrangement formed by genetic guidance and environmental input at that moment. Any responsiveness it exhibits is shaped by its current conditions rather than by memories carried across the transformation. In larger animals, memory persists because the physical structures that store it endure across years. Neurons remain in place. Synapses are maintained and modified rather than destroyed wholesale. In the hydrozoan cycle, however, the very act that enables renewal involves dismantling the tissue that could have preserved such continuity. The price of beginning again is the relinquishing of prior neural patterns. Thus, the organism's capacity to reset its life does not include the retention of lived experience. Its transformations favor structural renewal over continuity of perception. Each phase exists within its own present, responding to stimuli without reference to an earlier chapter. In this way, the cycle offers a biological fresh start, not only in form but also in neural organization, a quiet erasure that accompanies the remarkable return to youth, long before glass beakers and steel instruments shaped human experiments. The ocean was already conducting its own quiet trials. Within its vastness, gradients of temperature, pressure, salinity, and light shift across distances both immense and minute. Organisms drifting or anchored within these gradients encounter conditions that change subtly or abruptly, sometimes within a single tide. In such an environment, variation is not an exception, but a constant presence, and life responds through an ongoing process of adjustment. Marine ecosystems span extremes rarely found on land. Sunlit shallows give way to dim twilight zones, then to depths where light never penetrates. Coastal waters fluctuate with rainfall and evaporation, altering salinity in ways that challenge delicate physiology. Nutrient pulses rise from deep currents, briefly enriching surface layers before dispersing again. Each of these factors creates selective pressures that differ from place to place and from season to season. Organisms that persist here must either endure instability or exploit it. The ocean's fluid nature contributes to this evolutionary experimentation. Unlike terrestrial habitats bounded by mountains or deserts, marine environments are connected by currents that transport larvae, spores, and microscopic life across great distances. A lineage arising in one region can be carried elsewhere, encountering new combinations of temperature and chemistry. Some arrivals fail to establish themselves, while others adapt, their genetic variations interacting with unfamiliar conditions. Over generations, these interactions shape traits that may seem extraordinary when viewed in isolation. Simple body plans are common among marine invertebrates, and simplicity can permit flexibility. Fewer tissue types and less rigid structural constraints allow certain organisms to explore developmental pathways that might be inaccessible in more complex forms. In this context, traits such as extensive regeneration or life cycle variation can arise without destabilizing the organism's integrity. The sea hosts countless such forms, each representing a solution refined by its particular niche. Chemical diversity further enriches this experimental landscape. Marine organisms produce compounds for defense, communication, and competition, many of which have no direct counterpart on land. These biochemical innovations reflect the selective pressures of crowded reefs, shifting sediments, and open water where visibility is limited. Within such environments, survival often depends on subtle, molecular interactions as much as on physical strength. Isolation also plays a role. Certain marine habitats, such as enclosed bays or deep-sea vents, function as natural laboratories separated from surrounding waters by physical barriers or extreme conditions. Populations confined within them may evolve along distinct trajectories, accumulating unique traits over time. When circumstances change and connections reopen, these traits can spread, contributing to the broader diversity of ocean life. The hydrozoan, whose life cycle can reverse, emerged within this expansive field of variation. Its capacity for renewal is one expression among many of the sea's evolutionary creativity. The ocean does not design with intention, yet its dynamic conditions foster experimentation on scales that span millennia. Traits are tested against currents, predators, and shifting chemistry, retained if they confer advantage, discarded if they do not. Thus, the sea functions as a vast and continuous laboratory, not in the controlled sense of human research, but as an arena where countless biological possibilities are explored. Within its waters, organisms display forms and functions that stretch conventional expectations, each shaped by the interplay of environment and inheritance. The small voyager that renews itself is part of this broader tapestry, a product of marine experimentation unfolding quietly across epochs. Temperature moves through the ocean like an invisible tide, rising not in waves, but in fractions of degrees that accumulate year by year. Marine organisms sense these shifts at scales far finer than human perception. Proteins fold differently under warmer conditions, metabolic rates accelerate, and reproductive cycles adjust in response to altered seasonal cues. For small gelatinous animals whose bodies are composed largely of water, even slight warming can influence growth, timing, and survival. As global temperatures increase, surface waters in many regions have grown measurably warmer. In some coastal zones, heat persists longer into autumn and arrives earlier in spring. For certain jellyfish species, these changes can extend breeding seasons or hasten development from larval to adult stages. Warmer water may enhance metabolic processes, allowing faster growth when food is abundant. In such circumstances, populations can rise more quickly than before, forming blooms that alter local ecosystems. Yet, the relationship between warming and abundance is not uniform. Elevated temperatures can also stress organisms, especially if accompanied by reduced oxygen levels. Warmer water holds less dissolved oxygen, and coastal areas subject to nutrient runoff may experience hypoxic events where oxygen becomes scarce. Small hydrozoans, though resilient in some respects, remain sensitive to such chemical shifts. Prolonged exposure to unfavorable conditions can limit reproduction or increase mortality. Climate change also influences the distribution of plankton, the microscopic foundation of marine food webs. Altered current patterns and shifting temperature gradients affect where nutrient-rich waters rise to the surface. In regions where plankton productivity declines, jellyfish may struggle to find sufficient prey. In other areas, increased nutrient input from human activities can stimulate plankton blooms, indirectly supporting higher numbers of gelatinous predators. The outcome varies by location, shaped by a complex interplay of warming, circulation, and nutrient availability. As waters warm, species often expand toward higher latitudes, where temperatures were previously too cool. Records already show some marine organisms appearing farther from the equator than in past decades. For the hydrozoan capable of renewing its life cycle, such shifts may open new habitats. If transported by currents or human-mediated pathways into regions that have become climatically suitable, it may establish populations beyond its historical range. At the same time, areas that grow excessively warm may become less hospitable, leading to local declines. Ocean acidification adds another dimension. Increased carbon dioxide dissolves into seawater, lowering its pH. Although gelatinous organisms lack rigid calcium carbonate shells, acidification can influence broader ecosystems, altering prey availability and competitive interactions. Changes in the abundance of other species ripple outward, indirectly affecting small hydrozoans whose lives are entwined with the surrounding community. Climate-driven alterations rarely act in isolation. A region may experience warming, deoxygenation, and shifts in predator populations simultaneously. In some cases, jellyfish numbers increase where fish stocks decline, altering ecological balances. In others, environmental stress suppresses multiple species at once. The hydrozoan's unusual capacity for renewal may offer resilience against certain disturbances, yet it does not grant immunity to widespread environmental change. Thus, as the planet's climate continues to evolve, the distribution and abundance of this small marine organism are likely to change in turn. Some coastal waters may host it more frequently, while others may see it diminish. The patterns will reflect not a single cause, but the cumulative effect of temperature, chemistry, currents, and ecological relationships. Within these shifting conditions, the species will persist where it can, adjusting its quiet cycles to the altered rhythms of a warming sea. Across the surface of the world's oceans move vessels whose journeys trace lines between continents, carrying cargo, fuel, and passengers along routes repeated so often they resemble invisible highways. Beneath their decks, within dark tanks designed for balance and stability, seawater is taken in and released again as ships load and unload. This exchange, routine and largely unnoticed, has become one of the most effective means by which small marine organisms travel beyond the limits of natural currents. Within those enclosed reservoirs, minute creatures drift unnoticed. Larvae, spores, and tiny invertebrates drawn in with coastal water may survive the voyage, protected from predators and buffered against changing conditions. When ballast is discharged at a distant port, the water returns to the sea along with its living cargo. In this way, species that once occupied only a narrow range can appear thousands of kilometers from their origin, arriving not through their own movement, but through the pathways of human commerce. The small hydrozoan, known for its unusual life cycle, has likely benefited from such transport. Reports of its presence in widely separated regions suggest that shipping activity has helped extend its distribution. Ports with frequent international traffic often host communities of organisms not native to the surrounding coastline, their arrival tied to repeated introductions over years. Some of these newcomers fail to persist, unable to adapt to unfamiliar conditions. Others survive, reproduce, and gradually become part of the local ecosystem. When a species establishes itself in a region beyond its historical range, it may be described as invasive if its presence disrupts existing ecological relationships. Not all introduced organisms become invasive, yet the possibility remains whenever new arrivals compete with native species for food or space. The hydrozoan's small size and specialized feeding habits reduce its direct impact compared with larger invaders, though its spread still reflects the broader influence of global transport on marine biodiversity. Adaptability plays a quiet role in this expansion. The species tolerates a range of temperatures within warm seas and can persist under varying conditions as long as essential requirements are met. Its life cycle includes stages suited to different circumstances, allowing it to endure changes that might limit less flexible organisms. Such versatility increases the likelihood that individuals released into unfamiliar waters will find niches where they can survive long enough to reproduce. Ocean circulation continues to assist its dispersal once established. Currents carry offspring away from their point of origin, distributing them along coastlines or into neighboring seas. Over time, successive generations may appear in locations progressively farther from the original introduction site. This gradual spread does not require rapid movement, only persistence across many cycles of growth and reproduction. Human activity has accelerated these processes. Trade networks link distant harbors that once had little biological connection. With each voyage, the boundary between regional ecosystems becomes more permeable. Some transported organisms perish quickly, yet others endure, their presence a reminder that the ocean, though vast, is increasingly interconnected by routes shaped not by tides alone, but by the passage of ships. The hydrozoan's expanding distribution illustrates how biological traits and global movement intersect. Its capacity for renewal may aid survival during transit, while its tolerance of varied conditions supports establishment in new habitats. Together with the steady rhythm of maritime travel, these characteristics allow it to appear in waters far from where its lineage first arose, its quiet cycles unfolding now across a widening portion of the world's seas. Duration in living systems is rarely absolute. Even processes that appear endlessly repeatable often unfold within boundaries that are subtle and difficult to observe. The small hydrozoan whose life can return to an earlier stage has demonstrated in controlled studies that its cycle may occur more than once. Individuals, kept under stable laboratory conditions, have undergone transformation, resumed development, and later repeated the sequence again. Such observations suggest that the underlying mechanism is not a single-use phenomenon, but a capacity that can be activated multiple times when circumstances permit. In these settings, researchers have monitored specimens across successive reversals, noting that the structural reorganization proceeds with similar stages each time. Cells alter identity, tissues rearrange, and a new phase emerges from the prior one. The process can unfold again after further growth, indicating that the biological machinery responsible for transformation remains functional beyond a single iteration. This repeatability has contributed to the species' reputation as biologically immortal, a phrase that refers to its resistance to intrinsic aging rather than to literal, endless existence. Yet laboratory observations do not fully capture the complexity of natural conditions. In the open ocean, countless variables influence survival. Temperature shifts, chemical fluctuations, scarcity of food, and encounters with predators all affect whether an individual lives long enough to attempt another cycle. Even if the internal mechanism can operate repeatedly, external circumstances may intervene before it is used many times. The difference between potential and outcome becomes especially important here. A capacity for renewal does not guarantee the opportunity to employ it indefinitely. Another factor lies hidden within the genome itself. Each time cells divide, they must copy their DNA. Although cellular repair systems correct many mistakes, some alterations inevitably remain. Over successive divisions, these small changes can accumulate. In most organisms, such mutations contribute gradually to aging or functional decline. For this hydrozoan, it remains uncertain how repeated transformations influence that accumulation. The rearrangement of tissues may redistribute cells, but it does not necessarily erase every genetic alteration. Whether mutations build up over many cycles or are mitigated by its repair mechanisms is still a subject of investigation. Scientists have begun to explore this question by examining genetic sequences from individuals at different stages and after repeated transformations. Early findings suggest that the species possesses efficient systems for maintaining genomic stability, yet no organism is entirely free from the laws of molecular chemistry. Radiation, reactive compounds, and ordinary replication all introduce the possibility of change. The long-term effects of these influences across numerous cycles have not been observed over spans that would reveal their ultimate limits. Time itself presents a practical challenge for study. Observing a single individual through dozens or hundreds of reversals would require conditions maintained consistently for years, perhaps decades. Such extended experiments are difficult to sustain, and so much of what is known comes from shorter observations. From these, it appears that the organism can repeat its transformation more than once, perhaps many times, but the precise boundary leading to eventual failure remains unknown. Thus, the question of endless repetition rests between evidence and uncertainty. The species demonstrates a remarkable ability to renew itself, supported by mechanisms that allow development to be revisited. At the same time, it exists within the universal constraints that govern all living matter. External hazards, molecular change, and the passage of time continue to exert their quiet influence. The cycle may turn again and again, yet whether it can do so without limit is a matter that nature has not fully revealed, leaving its true duration suspended between possibility and the unmeasured horizon of biological time. Inevitability in nature rarely arrives as a single event. Instead, it approaches through many small pathways, each shaped by chance and circumstance. The small hydrozoan, whose life can renew itself, remains subject to these pathways, its unusual biology offering resilience against internal decline, yet no shield against the wider forces that govern survival in the sea. Disease is among those forces, though it often goes unseen. Marine microorganisms surround every drifting creature, invisible companions that may coexist peacefully or, under certain conditions, become harmful. Bacteria, viruses, and protozoans can invade delicate tissues, disrupting cellular processes and weakening structure. In organisms composed largely of soft material, such infections may spread rapidly. A body capable of reorganizing itself cannot always outpace a pathogen that multiplies within its cells. When infection overwhelms repair, the cycle of renewal may end before it can begin again. Predation remains another constant presence. Countless animals feed opportunistically, consuming whatever nourishment they encounter. For a tiny gelatinous organism, an approaching predator may be detected only moments before contact. Some hunters rely on vision, others on touch or chemical cues, yet all share the same fundamental need for energy. When capture occurs, digestion proceeds without regard for the remarkable capacities of the prey. The internal chemistry that allows transformation cannot function once tissues have been broken down and absorbed into another body. Chemical hazards also shape survival. Coastal waters can carry substances introduced through natural processes or human activity. Heavy metals, agricultural runoff, petroleum residues, and synthetic compounds disperse through currents, sometimes concentrating in particular regions. Even trace amounts may interfere with cellular metabolism or disrupt signaling pathways. For organisms with permeable tissues, exposure can occur quickly. Molecules dissolved in the surrounding water pass across membranes and enter internal compartments, where they may alter enzymatic reactions or damage genetic material. Such effects do not always produce immediate death, yet they can weaken physiological stability until the organism can no longer sustain itself. Physical accidents contribute as well. Shifting sediments stirred by storms can bury small creatures or abrade their tissues. Sudden turbulence may carry them into unsuitable conditions, including areas where temperature or salinity lies outside their tolerance. Collisions with debris or entanglement in suspended particles can impair movement and feeding. These events are not rare anomalies, but ordinary features of a dynamic marine environment. Each represents a moment when circumstance overrides biological potential. The presence of these hazards illustrates a simple truth: biological immortality, in the sense of avoiding age-related decline, does not remove an organism from the web of interactions that define existence. Life persists only while structure remains intact and function continues. When external forces disrupt that integrity beyond repair, the process ends, regardless of how flexible the underlying biology may be. Even so, the species continues, because not every individual encounters such endings at once. Some survive infection, evade predators, or drift clear of contaminated waters. Their persistence allows the lineage to endure despite constant losses. Death, in this context, does not signify failure but participation in the same cycle that sustains all ecosystems, where energy passes from one form to another and matter returns to the surrounding environment. Thus, the small voyager remains both extraordinary and ordinary. Its cells hold the capacity to begin again, yet its existence is still bounded by the same uncertainties that govern all living things. Renewal can delay certain endings, but it cannot abolish them. Within the shifting conditions of the sea, life continues only for as long as circumstance permits, and when circumstance changes, even the most remarkable biology yields to the quiet finality shared by every organism. Names often arrive before understanding settles into place. A phrase can drift through headlines and conversations, gathering brightness as it travels until it seems larger than the quiet biology beneath it. The word immortal has followed this small marine organism in that way, attaching itself firmly to a life cycle that bends backward against expectation. In scientific language, immortality does not mean invulnerability. It does not imply endless survival through every storm or predator encounter. Instead, it describes the absence of senescence, the gradual internal decline that most organisms experience as cells accumulate damage and systems lose efficiency. For this hydrozoan, laboratory observations indicate that its tissues can return to an earlier developmental state rather than progressing toward irreversible aging. Its cells avoid the typical pattern in which time steadily narrows biological possibility. When researchers describe it as biologically immortal, they are referring to this capacity to reset development. The organism can, under certain conditions, transform from a mature medusa back into a polyp stage, effectively restarting its life cycle. In doing so, it sidesteps the terminal deterioration that defines aging in many animals. The phrase is precise within that context, yet outside scientific circles, it often expands beyond its intended boundaries. Media accounts have sometimes presented the species as incapable of dying, as though it drifts through centuries untouched by hazard. Such portrayals simplify a far more nuanced reality. The organism remains vulnerable to predation, disease, environmental stress, and countless unpredictable events. Its immortality lies not in immunity to harm, but in its unusual relationship with time. It can avoid one specific form of death, the one that arrives gradually from within. The distinction is subtle yet essential. Most animals follow a largely unidirectional developmental path. Growth leads to maturity, maturity to reproduction, and reproduction to decline. Even when lifespans vary widely, the arc itself remains fixed. This hydrozoan interrupts that arc. Instead of moving steadily toward a final stage, it can loop backward, revisiting earlier configurations. The terminology of immortality attempts to capture that departure from the usual trajectory. Scientists remain careful in their phrasing because biological systems rarely operate in absolutes. Even the capacity for reversal may have limits not yet fully measured. Genetic mutations can accumulate, environmental pressures can interfere, and energy constraints may restrict how often transformation occurs. Within laboratories, repeated cycles have been observed, yet no experiment has followed a single individual across indefinite spans. The adjective immortal therefore describes a property, not a guarantee. Language shapes perception, and perception shapes expectation. When a creature is labeled immortal, it seems to challenge the very framework of life and death. In reality, it challenges only one aspect of that framework. It reveals that aging, as commonly understood, is not universal. Some organisms possess developmental flexibility that alters how time influences their bodies. This flexibility does not free them from ecology or physics, but it expands the range of possible life histories. Thus, the name persists, luminous and slightly misleading, pointing toward a genuine biological marvel while inviting careful interpretation. Immortal, in this context, signifies resistance to intrinsic aging, not escape from mortality itself. It is a reminder that nature contains variations more intricate than familiar categories suggest. Within the quiet cycles of the sea, a small organism demonstrates that life need not always proceed in a straight line toward decline, and that even a single exception can reshape the way we speak about time and survival. Time in living systems is often imagined as a quiet current, carrying cells and bodies from emergence toward conclusion. Within that current, biological clocks measure progression in subtle increments. Telomeres shorten as cells divide, proteins accumulate, small errors. Metabolic byproducts leave faint traces in tissues. These processes form a rhythm that, in most organisms, moves in one direction only. Growth leads forward, aging follows. The arrow rarely bends. Across species, this directionality appears almost universal. From insects that live for days to trees that endure centuries, life advances along a developmental path that does not reverse itself. Even regeneration, remarkable as it may be, usually restores lost structure without altering the fundamental sequence of age. A salamander may regrow a limb, yet it does not return to infancy. The clock continues its steady counting, indifferent to repair. The small marine organism that has accompanied this story unsettles that expectation. Its cells demonstrate that the biological clock, at least in certain forms of life, is not entirely rigid. Under specific conditions, mature tissues reorganize into earlier developmental states. The progression that once seemed linear reveals the capacity for curvature. Instead of moving only toward later stages, the organism can revisit beginnings, as though the current of time briefly loops back upon itself. This phenomenon does not abolish time. Rather, it exposes the mechanisms through which time exerts influence. Aging is not a mystical force, but a collection of molecular events. When telomere shortening, oxidative stress, and DNA damage accumulate beyond repair, function declines. In this hydrozoan, robust repair pathways and cellular plasticity alter that accumulation. Developmental identity becomes flexible. The clock does not stop, yet its hands may reset to an earlier configuration. Such flexibility challenges the assumption that directionality is fixed for all life. It suggests that what appears inevitable may in fact be contingent on structure and scale. Complex animals rely on tightly integrated organ systems and specialized cells that resist change. In simpler bodies, composed of fewer layers and distributed networks, transformation may be less disruptive. The constraints that bind one lineage need not bind another. Even so, the exception remains rare. Among the vast diversity of life, only a handful of organisms display comparable abilities. The overwhelming pattern still favors forward progression. Evolution, guided by reproduction and survival within ecological contexts, does not universally reward indefinite renewal. In many environments, investing energy in rapid growth and reproduction proves more advantageous than maintaining perpetual youth. The arrow of life persists because it serves a functional balance. The hydrozoan therefore teaches not that time is illusory, but that its biological expression can vary. The organism inhabits a narrow margin within possibility, demonstrating that developmental sequences can be rearranged when genetic architecture permits. Its existence expands the conceptual boundaries of aging without overturning the broader order of nature. In contemplating this rare deviation, one sees time less as an unyielding river and more as a pattern shaped by underlying rules. Those rules may allow, in exceptional cases, a return to earlier states. Yet the return does not erase exposure to environment, nor does it confer dominion over mortality. It simply reveals that life's chronology is more intricate than a single straight line. Within the quiet waters where this organism drifts, the lesson remains understated. Time continues, currents flow, and most creatures follow their unidirectional path. Amid them moves a small reminder that biological clocks are constructed systems, and that under certain conditions, even the direction of life can momentarily soften, curve, and begin again. It is easy to imagine that a creature capable of bending its life cycle must possess some visible grandeur, a mark of its rare capacity written boldly into its form. Yet the body that carries this unusual trait is almost weightless in appearance, scarcely larger than a fingernail, nearly transparent against the shifting light of shallow seas. Its bell is delicate, its tissues thin, its movements subtle. The very organism that unsettles our ideas of time exists in a shape so fragile that it can vanish from sight with the slightest change in angle. Transparency serves both as camouflage and as vulnerability. Light passes through its tissues, revealing only faint outlines of internal structures. In clear water, this quality makes it difficult for predators to detect, allowing it to drift with a measure of quiet concealment. At the same time, such a body offers little physical protection. There are no hard shells, no thick armor, no rigid defenses. Its architecture is composed primarily of water held together by soft matrices of cells. A sudden compression, a careless bite, or a turbulent surge can disrupt that arrangement with ease. The small scale of its existence shapes every aspect of its survival. Nutrients must be captured in microscopic quantities, suspended in the surrounding water as planktonic life. Feeding depends on delicate tentacles that extend outward, armed with stinging cells too small to see without magnification. These structures are effective within their scale, yet they operate in a world where larger mouths constantly sweep the same waters. The balance between concealment and exposure remains precarious. Being easily overlooked carries its own paradox. For scientists, the organism's minute size once delayed recognition of its extraordinary cycle. Countless individuals likely drifted unnoticed, completing transformations beyond human awareness. Even now, observing them requires careful collection and patient study. In the wild, their lives unfold quietly, unmarked by spectacle. Their looping pattern of development does not announce itself. It is embedded within tissues that appear unremarkable at first glance. Fragility also characterizes the process of reversal itself. When conditions deteriorate, the medusa contracts and begins the intricate reorganization described earlier. During this period, its structure becomes even more vulnerable. Tentacles retract, the bell shrinks, tissues collapse inward. The organism forms a cyst-like stage that adheres to a surface. In that compressed state, survival depends on stability. A disturbance at the wrong moment could end the sequence before renewal is complete. The capacity to begin again exists alongside the risk of interruption. Such delicacy underscores a broader truth about exceptional traits in nature. They do not always appear in dominant or imposing forms. Sometimes they reside in bodies that seem almost incidental, as though evolution experimented quietly within a narrow margin. The hydrozoan's looping life does not grant it size, strength, or ecological supremacy. It grants flexibility within a specific context, a subtle adjustment to the pressures of its environment. In the vastness of the ocean, where currents stretch across continents and storms reshape coastlines, this organism persists as a near invisible participant. Its transparency allows it to blend with shifting light, yet that same transparency reveals how little separates it from dissolution. The tension between resilience and fragility defines its existence. It can revisit earlier stages of life, yet it remains composed of materials that yield readily to force. Thus, the looping life it embodies is not a triumphant circle carved in stone, but a fine thread traced through water. It curves back upon itself when conditions demand, then extends forward once more, always subject to interruption. Within its tiny, translucent form lies a reminder that even the most unusual biological capacities can inhabit bodies of extraordinary delicacy, where endurance depends as much on invisibility and chance as on the remarkable machinery of renewal. Some lives are best imagined not as lines but as shapes. A line begins, extends, and eventually stops. A circle, by contrast, contains no final point, only continuous return. The small marine organism that has guided this exploration embodies such a circular pattern, its development capable of folding back upon itself when conditions require. In theory, this capacity suggests a life that need not conclude through internal decline. Its cycle can repeat, tracing the same path again and again, each turn restoring earlier form. This possibility has led many observers to describe its existence as endless. Yet in practice, continuity depends on more than internal design. A circle drawn on paper remains unbroken because nothing disturbs it. In the living world, every organism moves within surroundings shaped by chance, motion, and interaction. Currents shift, predators roam, chemical balances change. Under these influences, even a life capable of renewal may be interrupted. The cycle contains no built-in ending, yet it unfolds within a setting where endings are always possible. The distinction between theoretical immortality and lived survival rests in this difference. Internally, the organism's biology provides a mechanism that can avert senescence. Cells can reorganize, development can begin anew, and structural decline can be reversed. Externally, however, it shares the same vulnerability as any drifting creature. Its future is shaped by encounters, by the chemistry of the surrounding water, by events that arrive without warning. The circular design offers opportunity rather than certainty. Within calm conditions, the cycle may proceed quietly. Growth leads to maturity. Maturity may lead to reversal, and reversal to growth once more. Each phase connects to the next, forming a sequence that loops rather than terminates. Observed in isolation, such a pattern appears almost timeless, as though duration itself had softened its grip. The organism becomes a living illustration of continuity, sustained through transformation. Yet the sea is not an isolated setting. It is a domain of motion and interaction, where every life intersects with others. A passing fish, a shift in temperature, a trace of dissolved substance carried from afar can alter the course of a single existence. When such events intervene, the circle is not completed, it is interrupted. The capacity for renewal remains part of the organism's nature, but the opportunity to enact it has passed. This interplay between potential and circumstance reveals a broader truth about living systems. Biological traits define what an organism can do, not what it must experience. Possibility and reality do not always coincide. The hydrozoan's unusual cycle expands the range of its possibilities, allowing it to revisit earlier stages instead of aging in a single direction. At the same time, its reality is shaped by conditions beyond its control. Seen from this perspective, its life is neither endless nor fleeting but conditional. It may continue through many transformations if surroundings permit. It may cease after a single stage if circumstances shift. The circle exists as a design within its cells, ready to be traced whenever the moment allows. Whether that tracing continues depends on factors outside the design itself. Thus, its story offers a gentle reframing of what it means for a life to end. For this organism, conclusion does not arise from an internal clock reaching zero. It arrives when something interrupts the pattern. Until that interruption occurs, the cycle remains open, capable of turning once more, its path defined not by a fixed destination, but by the quiet possibility of return. At the far edges of perception, where observation fades into patience, the natural world often keeps its most intriguing departures from expectation. The small hydrozoan that has accompanied this journey does not proclaim its difference through color or motion. It drifts, nearly invisible, its presence easy to miss. Yet within that modest form resides a pattern that quietly unsettles one of biology's most familiar assumptions, the idea that every life must proceed along a single, unbroken path from beginning to end. The discovery of its unusual cycle did not arrive as a thunderclap revelation. It emerged through careful attention, through scientists noticing a transformation that at first seemed improbable. Repeated observation confirmed that the phenomenon was real, not illusion or error. Each confirmation deepened understanding, while also widening the sense of how much remains unknown. Such moments illustrate the nature of scientific progress. Knowledge grows not through certainty alone, but through the willingness to question what appears settled. This organism's capacity for renewal does not overturn the principles that govern life. Physical laws remain unchanged, ecological pressures continue, and mortality still shapes existence. What it offers instead is a reminder that those principles allow more variation than our assumptions sometimes anticipate. Within the boundaries of chemistry and evolution, there is room for patterns that seem almost improbable until they are witnessed. Its story also reflects the importance of scale in shaping perception. Large animals often draw attention because their forms are easy to see and their movements easy to follow. Smaller creatures pass unnoticed, their lives unfolding beyond casual observation. Yet it is often among these overlooked beings that unexpected biological strategies appear. Their simplicity can permit forms of flexibility that complex bodies cannot easily sustain, revealing possibilities that broaden the map of life's designs. The ocean, vast and layered, continues to serve as a reservoir of such discoveries. Beneath its surface lie habitats shaped by pressure, darkness, and shifting chemistry, each supporting organisms adapted in ways that may seem unfamiliar to those who study life primarily on land. With each exploration, researchers encounter structures, behaviors, and biochemical pathways that expand the catalog of what living systems can accomplish. The hydrozoan's looping development is one thread in that larger tapestry, a subtle variation woven into the immense fabric of marine diversity. Scientific humility grows from encounters like this. Each new finding carries the suggestion that understanding is provisional, that even well-established ideas may contain room for revision. The recognition that a tiny organism can revisit earlier stages of life encourages a gentler view of certainty. Rather than closing questions, it invites them, not as challenges to be conquered, but as openings through which knowledge may continue to flow. In the quiet rhythm of its transformations, this small voyager leaves an impression not through spectacle, but through implication. It suggests that nature's creativity extends beyond the patterns most often observed, and that exceptions can illuminate rules more clearly than conformity alone. Its existence does not demand astonishment. It simply stands as evidence, delicate and persistent, that life possesses a capacity for variation deeper than habit might suggest. So the story comes to rest where it began, with a creature almost too small to notice, moving gently through its world. It carries no proclamation, no visible emblem of its rarity. Yet within its translucent body lies a lesson shaped by time, patience, and observation. The largest assumptions about life are sometimes challenged not by giants or extremes, but by the quiet presence of something easily overlooked, continuing its cycles in silence.